UELC 5 New Trends in Pharmaceutical and Biological Sciences by Yoel Dagan, Laure Kindele, and Rima Chatila. Introduction. This presentation is about the TRX and TXNIP system. The aim of the study is to point out the different key roles of that couple in our cells, and another purpose is to determine whether TXNIP could be used as a therapeutic target in ischemia. Let's start with some general information about the first article. This one is titled Role of Endothelial TXNIP in Vascular Aging and Post-Ischemic Angiogenesis. It has been written by Alison Domingue and published in 2018. It's an overview of different studies that have been conducted so far about the TRX and TXNIP system. Presentation and structure of the TRX and TXNIP system. TRX, also known as thyroidoxin, is an antioxidant enzyme present in all living cells. It catalyzes the formation of disulfur bonds between cysteine residues within proteins at their fault. TXNIP is also known as a thyroidoxin interacting protein. Several studies have highlighted the key role of TXNIP as an inhibitor of TRX. As a result, TXNIP modulates the effect of TRX, creating a balance between oxidant and antioxidant effect. TXNIP as a transport protein. TXNIP's arrestin-like structure suggests that it is also a transporter. In fact, TXNIP can carry TRX to the membrane in response to reactive oxidant species. Another study revealed that TXNIP may promote TRX accumulation in the cardiomyocyte's nucleus, protecting from cardiac hypertrophy. TXNIP's regulation. TXNIP's expression in vitro is increased by several factors, such as carcinogenic factors, stress, metabolic conditions, and thermic shocks. More recently, several microRNA were used to regulate TXNIP's expression in cells. Overexpression of microRNA 135 and 128 during ischemia resulted in the decrease of TXNIP in cells. In contrast, hypoglycemia, which reduces level of microRNA 128, is likely to raise TXNIP's expression. Pharmacological inhibition of TXNIP. The following section is a review of several studies about TXNIP targeting drugs and their effects on various cells. Oral antidiabetics are known to decrease TXNIP levels in cells. Thus, metformin, the drug leader of antidiabetics, have been used to reduce TXNIP in cancerous cells. Hypocholesterolemians, such as rosuvastatin, alleviate inflammation by reducing TXNIP levels. Calcium channel blockers, such as verapamil, have the same effects on TXNIP as the drugs before. And finally, aspirin, as an NSAID, inhibits reactive oxygen species accumulation by reducing TXNIP's expression in cells. TRX and TXNIP's impact on metabolism. TXNIP is a key protein when it comes to metabolic regulation. Some of the studies showed that TXNIP promotes the GLUT1 transporter's internalization and downregulation, causing troubles in the glucose pathway. Assuming that TXNIP is overexpressed in metabolic conditions such as diabetes, several studies identified this protein as a potential target. A study revealed that mice whose TXNIP gene was silenced couldn't survive long fast and developed several metabolic conditions, such as hypoglycemia, hyperketonemia, and hypertriglyceridemia. It also reduced the risk of developing insulin resistance when fed with a high-fat diet. TRX and TXNIP's role in inflammation. TXNIP is also involved in the inflammatory pathway. By reacting with the NLRP3 receptor, TXNIP amplifies the inflammation in endothelial cells. In fact, both the inflammasome and caspase-1 are triggered by NLRP3, leading to the synthesis and release of IL-1 beta, an actor of inflammation. Under physiological conditions, TRX and TXNIP are bound together, avoiding any possible contact between TXNIP and NLRP3. Apoptosis, proliferation, and cell survival. TRX binding to ACT1 results in an anti-apoptotic effect. On the contrary, TXNIP inhibits TRX binding to ACT1, promoting cell apoptosis. When high blood sugar levels are detected, TXNIP acts as a pro-apoptotic factor, promoting endothelial cells death. The induction of TRX by PDGF is known to promote cell proliferation. On the other hand, TXNIP inhibits cell proliferation by trapping TRX in the nucleus. TRX, TXNIP, and cell mobilization in bone marrow. TXNIP's function in bone marrow differs a lot from the previous findings. Investigations on murine cells showed that TXNIP altered the synthesis of reactive oxygen species in hematopoietic cells, which makes it an antioxidant. Moreover, silencing TXNIP revealed higher levels of reactive oxygen species and a higher risk to undergo apoptosis. In addition, TXNIP promotes stem cell quiescence by enhancing the communication between stem cells and their niches. A study revealed that hematopoietic cells mobilization depends on AKT phosphorylation, which TXNIP inhibits. This way, TXNIP inhibits hematopoietic cells migration. TRX, TXNIP, and angiogenesis. TXNIP mediates the translocation of TRX to the membrane when physiological levels of reactive oxygen species are detected. TXNIP is therefore necessary to TRX antioxidant function in such circumstances. TXNIP allows TRX to interact with growth factors, such as VEGF, followed by a pro-angiogenic function. Curiously, under hypoglycemic conditions, TXNIP seems to have the opposite effect. It reduces levels of VEGF, resulting in an anti-angiogenic response. These statements were confirmed while using a mutant of TXNIP unable to bind and interact with TRX. It means that TXNIP anti-proliferating properties work independently of TRX. Moreover, silencing TXNIP seems to re-establish the cell's response to VEGF. TXNIP anti-angiogenic role is mediated by the HIF-A transcription factor, to which TXNIP usually binds. The interaction between both factors prevents HIF-A from translocating in the nucleus. It leads to HIF-A's inhibition by the cell proteasome in the cytoplasm, along with its pro-angiogenic functions. TRX and TXNIP in ischemia. In a study about cardiac ischemia, researchers showed that NLRP3 inflammasome levels are increased during ischemia. This is a result of TXNIP overexpression in such case. This inflammation process is a major cause of post-ischemic dysfunction. It can indeed lead to ventricular remodeling and cardiac insufficiency. Targeting NLRP3 with a microRNA resulted in the decrease of cardiac lesions, while targeting TXNIP with a specific microRNA reduced the inflammation. This suggests that both TXNIP and NLRP3 could be potential targets in the future. On the contrary, the injection of TRX vectors in diabetic rats restored cardiac function after ischemia. Moreover, resveratrol, a drug that increases TRX dose dependently, improves cardiac reperfusion and protects the heart from post-ischemic lesions and disabilities. Lower limb ischemia and strokes. A team of researchers injected anti-TXNIP factors, such as fenofibrat, in the lower limb of rats suffering from ischemia. It resulted in a better vascularization of the lower limb, and it also reduced the bad effects of a high-fat diet after lower limb ischemia. On the other hand, another drug, omaliferone, was able to reduce TXNIP level in brain, and thereby attenuate brain damages after a stroke. Targeting TXNIP also seemed to protect neuronal cells and increase their viability. Alright, as a quick recap, the TRX and TXNIP system is a couple of proteins involved in many functions of the organism. TRX is an antioxidant enzyme present in all living cells. It reduces inflammation and promotes cell proliferation. It is also a pro-angiogenic factor since it is able to restore perfusion of an ischemic site. TXNIP inhibits TRX, which makes it an antioxidant. TXNIP is better expressed in critical circumstances such as cell stress, cancers, and metabolic diseases. It is a pro-inflammatory and anti-angiogenic protein. During ischemia, silencing TXNIP seemed to lower damages and increase cell viability, which makes it an interesting target in the future.